you guys believe it, we've made it to the very last chapter of your textbook. So chapter 29, its topic is genetics and hereditary traits. As you guys probably understand by now, genes are important for our DNA, our blueprints, which, remember, are synthesizing plans for proteins. So we should know at this point that the bulk of our body is made up of protein. So we have skin, we have hair, we have nails. All of these things are built of protein. DNA is what codes for that specific formation of proteins. Remember, we have the DNA inside of our cells that codes for a specific protein. That protein will be realized by using messenger RNA, which then goes through the process of transcription, translation, in order to form polypeptides within our cells. If you guys don't remember that, go back to chapter 3 and take a look at it. So the question then is, what is genetics? So genetics is the study of how things are passed hereditarily. It was first studied, well, it's been studied for a long time, but it was first really proposed and first realized by Mendel. So Mendel was said to be the grandfather of genetics. He studied inherited characteristics, and the way that he did this was through a concept of all or none. And we've since realized since the 1800s. Now remember, he was doing this before electricity was even invented. So it's pretty amazing the work that Mendel did. Since then, we've created the Human Genome Project, which has determined the exact human DNA sequence, which we know now we can genetically map our DNA, which can aid in screening for certain diseases and whatnot. So we need to learn about vocabulary of genetics. So we've kind of talked about this in the last chapter, that the word diploid or diploid versus haploid. Okay, so what do these words mean? So diploid refers to the number of chromosomes, 46, which humans have in all of their cells, except for the gametes, which only have 23. So I'm just going to put body cells here and gametes here. And of these cells, we say 46 is 23 pairs. So 23 times 2, 23 pairs of what are called homologous chromosomes. So we got one pair, one side of that pair, 23 from our mother, and we got 23 from our father. Out of these 23, 22 are said to be autosomes, and one is the sex chromosome, which is where we get XX and XY. So one pair is the sex chromosomes, and 22 pairs are the autosomes. Those are the things that determine all of our genetic traits. When we look at the vocabulary of genetics, you see in your text and in your book karyotype. Okay, so karyotype is when you actually put chromosome number 1 from your mother next to chromosome number 2 from your father, and so on and so forth. You can actually look at figure 29.1 in your text. If you have your textbook, go ahead and pull it out. Pause this right now. Pull out your textbook. Turn to figure 29.1 because we'll be looking at a few pictures in this presentation. So if you look at this particular image, the slide that you see on the left, number 1 there, is how the chromosomes exist naturally. They're all just kind of interspersed. But then if you take each one with the computer and place it next to each other, you'll find a karyotype in number 3. So karyotype is a displaying of homologous pairs. Genome is the genetic makeup. We got one set from our mom and one set from our dad, like we said. So then we look at what are called alleles. Okay, alleles are important because this is what codes for a specific thing. So alleles are genes that occur at the same location on a pair of homologous chromosomes. Okay, so if this allele codes for the same trait, it's said to be homozygous. Okay, homozygous. If it codes for different traits, if the allele codes for different traits, it's said to be heterozygous. Okay, so these two terms are going to be important. AKA, if you have one allele that encodes for blue eyes on one side of a pair of chromosomes, and the other side codes for brown eyes, that is said to be then a heterozygous pair. Now also when we talk about alleles, it's important to note the concept of dominance. Okay? So when we have dominance, one allele will suppress the other allele, which is said to be then recessive. So we have dominant and recessive. Oh, I'm spelling that wrong. That's just one C there. Boop, recessive. The letters are typically denoted by the same letter but using a capital versus a lowercase letter. So if we were going to use, say, the eyes, we would say the capital B codes for brown eyes, whereas the lowercase b codes for blue eyes. And in this case, brown is dominant over blue. So then we would have three scenarios for possibilities. Okay, these are the three scenarios. So since you have a homozygous dominant case here, okay, that's going to result in brown eyes. This would be a heterozygous case because you have two different alleles, and what would be the resulting of this would be brown again. Why? Because the dominant masks the recessive, and then over here this would be homozygous recessive case, where this would result in blue eyes. Okay, so just to recount then, this would also be a brown-eyed case because you have two homozygous dominant alleles. So what I have just written here is the genotype and the phenotypes. The genotype is writing it on paper. What does the genetic makeup actually look like, letter-wise? This is the phenotype. Phenotype means if I were to look at this person physically with my eyes, would I see brown eyes on them or would I see blue eyes? So make sure you know what genotype and phenotype mean. For independent assortment, what this means is that the alleles of two different traits are on two different chromosomes, and they're distributed independently of one another. So for example, in your text you see that capital B, lowercase b for hair color is on one chromosome, while capital J and lowercase j for double thumb jointedness is on another chromosome. So the typical possibilities are a combination of those four letters. So inheriting capital B or lowercase b versus a capital J or a lowercase j would create four different possibilities for two chromosomes. So what we see is the number is actually defined as 2 to the n power. Therefore, if you had two chromosomes, that would be 2 to the second power, which would equal 4. If you had four chromosomes, that would be 2 to the fourth power, which would be 2 times 2 times 2 times 2, so that's 2, 4, 8, 16. So if we extrapolate this down and see 23, because we have 23 pairs of chromosomes, that is 8.5 million combinations. So that's 8.5 million combinations just for the gametes alone. And that's only number one of this three different factors for genetic variation. So for part number 2, I want you to look in your textbook. So if you have your textbook, pause right now and find this picture. Figure 29.3-1. So what we see in this particular image is homologous chromosomes. They've synapsed during prophase of meiosis 1. Each chromosome consists of two sister chromatids. So these are considered the chromatids, two sister chromatids. Okay, so we see here that the location for hair color gene is on this particular allele, and the allele for eye color is down here. Okay, so we see also that the purple is paternal and the green is maternal. All right. Now, notice the combination here, capital H, capital E. That's going to be brown hair, brown eyes. Capital H, capital E, same. Lowercase h, lowercase e, codes for blue hair, and blue eyes. Okay, so we see here those particular pairs. Now, during an event called crossover, what would happen is they don't particularly line up just straight like that. That's not what this is not what typically happens in real life. What happens is they cross over. So when they cross over now, we see that instead of lowercase h little e, now we have lowercase h capital E. So that when they separate, okay, the genetic material actually grabs onto whatever's closest to it, so that when they separate out, now we have different combinations that then what would have been formed if this E was back here in its original place. So this event, okay, causes, so this is called crossover and then genetic recombination. This is the recombination when they combine. Okay, this creates much genetic diversity. Now the third event, random fertilization, means that a single egg is fertilized by a single sperm in a completely random manner. Therefore, independent assortment, which was number 1, okay, plus the crossover, plus the random fertilization would result in approximately 72 trillion. And really this is not even taking into account the crossover, because if you just multiply 8.5 possibilities for the egg and 8.5 million possibilities for the female, multiply these together, that's where you get 72 trillion possibilities, and that's without crossover. So this is why no two people on Earth look exactly alike. Well, don't debate me unless you have an identical twin, that's different. But no two people who are unrelated or even brother and sister look exactly alike because of this genetic variation. Now, section 29.3 in your book details patterns of inheritance. So, phenotypes can be traced by a single gene, and most traits determined by multiple alleles, or by interactions of several gene pairs. We're going to use what's called a Punnett square. So, what you're looking at here is a Punnett square. You can draw this on a piece of paper if you like and practice with me. Okay, Punnett square is a diagram used to predict gene combinations from known statuses genotypically of parents. So for example, in your book they give an example of tongue rolling. So if I have a mom, so let's say maternal here, paternal here, it really doesn't matter which side you put them on because you're going to end up with the same results. So if you have a mother who is a tongue roller, but so let's first of all let's look at what tongue rolling is. So capital T's, combination of two. So these are our possible combinations where two capital T's, which remember we call this homozygous dominant, okay, homozygous dominant, they would be tongue rollers, whereas the heterozygous case would be tongue rollers as well, because the capital T dominates over the lowercase t. And in this particular case, this would be homozygous recessive. They would be non-tongue rollers. So if I have a case where a mother is a heterozygous tongue roller, and the father is also a heterozygous tongue roller, that would give 4 possible combinations. So you bring each letter in. Okay, let's bring this down and this one across. Same thing over here, bring him over, down, and then, so those are our 4 possible combinations for these two parents' offspring. So we have one possibility of homozygous dominant, one possibility of homozygous recessive, and then 2 possibilities of heterozygous. So if we were to express the results mathematically, we would say the ratio is 1 to 2 to 1, or 25% to 50% to 25%. Okay, and you can actually put in your results here. Okay, now this is what's considered, remember, a genotype ratio. What would the phenotype ratio be? Meaning, what would these people look like expressed-wise? How many would result in tongue rolling and how many wouldn't? So would this case result in tongue rolling? Yes. Would this case result in tongue rolling? Yes. Would this one result in tongue rolling? No. So how many is that then? So we have 1, 2, 3 that are tongue rollers versus 1 that is not a tongue roller. So the ratio of the phenotype would be different than the ratio for the genotype because you have to look at it expressed-wise rather than on paper. This is all statistical based. This is all probability. It is not actually determinate. It just shows you the probability that if a person were to have 4 children, you would think that each one would be expressed, okay, in each one of these 4 boxes. They could potentially have 3 children that are non-tongue rollers. That would be against the odds, but it is a possibility. Remember, this is all probability, statistical based. If you turn over in your textbook a little bit farther down, you're going to see table 29.1. This particular table shows you dominant genes and recessive genes determined by the inheritance. So you see things like astigmatism, things like pigmentation, certain diseases are even determined by dominant recessive inheritance. Now, there are cases where, and what we've been talking about so far is what's called complete dominance, where one trait is completely dominant over the other. Now, there are also cases where we have what is called incomplete dominance. Okay, incomplete dominance. Incomplete dominance is best seen in the body with a condition called sickling, so sickle cell anemia. And in this case, the heterozygous individual, okay, so the heterozygous individual would express something different than the normal heterozygous when we talked about like complete dominance. So if we look at the 3 cases for this particular topic, so we would have the 3 scenarios as before, okay, where this first person would express normal hemoglobin. The sickle cell anemia would be seen all the way at the bottom. So this is the person with full-blown sickle cell anemia. And so this is a person who only makes mutated hemoglobin, which would be someone who is very susceptible to sickle cell crisis in low oxygen situations, whereas the intermediate is called sickle cell trait. So both the mutated form and the normal form are created. So this person would sometimes have sickle cell problems, so express sickle cell crisis under a very prolonged reduction of blood O2. So here we see normal and mutated hemoglobin formed. So this is called incomplete dominance. In our next case, we have what is called multiple allele inheritance, which means that the genes that exhibit more than 2 allele forms. There's not just like capital T, lowercase t. And the one place we see this in the body is the blood typing. So we look at the ABO blood groups. You guys remember this from way back when? This was the first chapter we did this semester, ABO blood groups. There are 3 different alleles designated by the letter I. So we have a capital I with an A superscript, a capital I with a B superscript, and a lowercase I. Alright, so then we look at the possibilities for these 3 alleles. Now you can only still have 2 alleles, but now you have 3 possibilities. So if we look here at this chart, this is table 29.2, looking at the genotype column, okay, we have a specific case here of complete, I shouldn't say complete, I should say homozygous cases where the capital I with the superscript A or B is passed on, double dose, meaning they would result in A and B blood types. But if you also look, there's a heterozygous case as well. So heterozygous A or B with the capital I passed along with the lowercase I, the capital I with the superscript A or B would dominate over the lowercase I, thus resulting in the blood types A or B as well. So those are heterozygous cases that show up in the phenotype A and B. Now, at the top and the bottom of the chart, you see, okay, homozygous cases, or homozygous case for the lowercase I, that's going to result in the blood type O. And then you have a heterozygous case where they're both the dominant, so they share dominance in this case, and you result in A and B blood types. Also in this chart you see the frequency of U.S. population. We also saw this particular statistic way back when, seeing that the O is the most common blood type in America for people of different ethnic backgrounds. So let's do a little practice, because this will be on your final exams. If you have a case where this is one parent, and this is one parent, and the capital D, we'll say it's for Dimples, is dominant over the lowercase d, which is not dimpled, what would be the possibilities for the offspring? And also, what would the genotype and phenotype ratios be? So I want you to pause this PowerPoint, or sorry, show me, and I want you to write this down on a piece of paper. I want you to write down the Punnett square, and then I want you to give me the genotype and the phenotype ratios. Okay, pause it now because I will go over the answers next. So what you should have seen, or what you should have filled in, is the following. For your Punnett square. And then for your genotype ratio, you should have had 2 to 2, but really we call this 1 to 1. So when you look at statistics, 2 to 2 is the same thing as 1 to 1, meaning 50-50 chance of being either heterozygous or homozygous recessive. And so what will this look like on the person? Same thing. Phenotype ratio would be 1 to 1 for dimple versus non-dimple. Okay? So let's do another example. In this case, capital F will stand for freckles. Lowercase f will indicate the absence of freckles. So here we have two cases, two people having offspring who both have freckles, but they're both heterozygous. What would that result in? So draw the Punnett square again, and then also give me the genotype and the phenotype ratios. Pause now. Okay, so you would have the following results. Which would then give a genotype ratio of 1 to 2 to 1 and a phenotype ratio of 3 to 1. So check yourself and see if you did that right. Now with blood typing, it's going to be a little more complicated. So let's say we have a mother who has blood type A, but she's heterozygous. And let's say we have a father who has blood type O. Alright, so what would the results be? So we would do this the same way we did before. you would bring the letters in from both sides. Okay, so I'm just going to draw down here at the bottom of each square. This is going to result in blood type A, correct? This will result in blood type A. This is going to result in blood type O and O. So here we see the genotype and phenotype ratios are going to be 1 to 1, okay, for both of these. So 1 to 1 genotypically and 1 to 1, okay, phenotypically as well. So we see here that if a mother was heterozygous blood type A and father was blood type O, those would be the possibilities for their children. Okay, so now I want you guys to practice, so draw this out on your paper, please. I want you to say, okay, so let's say the mother is heterozygous and she's blood type A, and let's say the father is also heterozygous, but he's blood type B. So I want you then to give me the letters here, because this can translate into that for both of these. And then I want you to give me the possibilities. Again, go ahead and write down here the phenotype, just like I did, and then give me the ratios. Okay, pause now. Okay, so here's your answer. So heterozygous A means they're different. And we know that she's blood type A, so only one possibility for that, and that would be here. Heterozygous B would be the same, but with the letter B, such as this. So then we're going to combine the letters in the boxes like before. And if we then write at the bottom, this is going to be AB, this will be blood type A, this will be blood type B, and this would be O. So we actually see is that a mother and a father who have blood types A and B could potentially have a child who's blood type O, genetically. So let's go ahead and do the genotype and the phenotype ratios. So what do you guys think? It's actually 1 to 1 to 1 to 1, 4 different kinds. So each one is 25%. And that would be for the 4 genotypes. Now phenotypes also 1 to 1 to 1 to 1, A to B to O to AB. Isn't genetics fun, guys? Okay, the last part is sex-linked inheritance for this dominant recessive inheritance section. Sex-linked inheritance means that inherited traits are carried on the sex chromosome, so either the X or the Y. So the X chromosome bears over 1,400 genes, whereas the Y chromosome only carries about 200. So we see that most genes are found on X chromosomes. So most of the important things that we talk about are going to be related to X chromosomes. So X-linked recessive alleles. So we have X and Y for male, and we have X and X for female. So if there is an X-linked recessive versus dominant, so dominant versus recessive would be like that. Dominant, recessive. So if this was a X-linked dominant recessive case where the male, okay, only one possibility for the male, but whereas the female would have to have a double dose in order to express whatever that recessive trait is. Okay, so we see here that X-linked recessive alleles are always expressed in males because there is no Y counterpart. So females must have both X chromosomes in order to express an X-linked condition. So it can be passed from mothers to daughters, but it must also be passed on from the father as well. Okay, then we also have polygene inheritance. Polygene inheritance. Polygene inheritance results in an actions, results are the results of actions of several gene pairs. Okay, so more genes are involved, there will be more phenotypic variation. So we see that skin color, eye color, height, intelligence, metabolic rate are all examples of things in the body where polygene inheritance comes into play. So such as this, we have for skin, alleles for dark skin, A, B, and C. Alleles for light skin, capital or lowercase, A, B, and C. So if someone is really dark skinned, they would have A, B, and C. Someone who was a little bit lighter would be, oops, not that, hold on. Like this. Or maybe you have this. Or maybe you have this. So do we see what we see here is that there are varying degrees of shades of skin color because there are so many combinations of these letters. So remember, all of these code for dark and all of these code for light. And so we have many different possibilities from polygene inheritance. There's a picture in your book, figure 29.6, that actually shows at the top of the page first generation. So you have parents, then you have first generation offspring. So you have, in this particular image, you have a very light skinned person, the lightest skinned person possible, someone from, like, I don't know, Sweden. And then you have a very dark skinned person. Okay, someone from, say, I don't know, Kenya. And they had a child together. So that child is going to then have just moderately colored skin. So you'd have a combination of the three letters. And say this person got together with another person of a comparable, I'm really struggling here, comparable skin tone. Okay, and they had children. Now what this says in this particular image is that the second generation offspring could potentially be really light skinned. So what if all of the lowercase letters were passed on? It would actually be possible, but a very small chance, only 1 in 64, that this person could be really, really light. And on the opposite side, there's a 1 in 64 chance that the person could be really, really dark. But what's most likely to happen is that it's going to fall somewhere in between, which is going to be a moderate skin tone. So it's very interesting when you look at genetics in this way, because you would probably notice this person has these two people have a moderate skin tone, and they're going to have a child that looks really light. You wouldn't think that that's normally possible, but it is. So the last section of this PowerPoint, or Show Me, is going to talk about genetic screening. Genetic screening provides information and options for prospective parents. So people like to have routine screening, okay, for certain genetic disorders. And so there's two ways that we really do this. The first way is by a pedigree. So if you have your book and you look at figure 29.3, it's an actual drawing of generations and drawings of males, which are represented by a box, and females, which are represented by a circle, and shading, which also, you see here, pink shade is going to be an affected individual, whereas a unshaded would be a unaffected individual. So you can see here in the first generation grandparents, you have man and woman, so you see marriage of families. We see lines that go across, that means they're getting married, and you see lines that come down, that means they're offspring. So it's a way to trace genetic traits through generations to help predict future risks. Now we also have number 2, blood tests. So there are blood chemistry or DNA tests that can screen children for maybe the sickling gene, so or cystic fibrosis. There are several things that we use this for. So the first one is called an amniocentesis. Amniocentesis, and what this is it's a withdrawal of amniotic fluid, which then is tested for genetic abnormalities. Typically, testing takes several weeks, whereas chronic sorry, not chronic, chorionic villus sampling, CVS, not the drugstore. Okay, CVS, chorionic villus sampling, is done where a little piece of the chorionic villi, which is the little fingers of blood tissue, is clipped and suctioned and sampled. So if you look at, to keep turning, figure 29.8, it actually shows you the process. So amniocentesis is a needle that is put into the amniotic sac to withdraw fluid, whereas a CVS is going to be a suction tube through the cervix. In both cases, karyotype is going to be formed. Okay, one's a little bit more invasive, so the CVS is more invasive, but the timing, so you see here, the timing, amniocentesis versus chorionic sampling, several hours versus several weeks. So you see the CVS, even though it's more invasive, you typically get results faster than with the amniocentesis, which could take several weeks. So this brings us to human gene therapy. So this is obviously brought up much debate, okay, in our society about ethical, religious, societal issues as far as if we can find a defective gene, we now have the ability to inject a corrected, quote, corrected gene into the cell. So, you know, there's different debates that go on whether people feel like this is too much control over genetics or whether we should just let life take its course. So, with that being said, we are officially finished with this textbook. So, good job, guys. Study hard for your upcoming tests.